Hello, I will be talking about Gill-Associated virus disease in prawn. So this will be quite a short discussion since I haven't found that many resources for this specific disease, but let's get into it. So Gill-Associated virus disease is caused by Gill-Associated virus 2. It's often differentiated from yellowhead virus 1, and this is because they are from the same species, from the genus Ocavirus, family Ranivieridae, order Nidoviralis, except lahi lang sila og genotype. So, YHV1 is genotype 1, GAV2 is genotype 2. It may also be called yellowhead virus 2. So, GAV is an enveloped, rod-shaped, single-stranded RNA virus. So, ang mga differences po sa GAV2 and yellowhead virus 1 is that they might share most of the same symptoms, pero there are some differences like makakita ka og yellow parts sa head sa shrimp or prawn sa yellowhead virus rather than sa GAV. So, here are some species that may be affected by GAV. So, the first species is the one that is quite significant jud sa GAV, and then they may be naturally susceptible. Ang kaning uban is included lang because they have been experimentally infected, but kani yung black tiger prawn. This is one of the most cultivated aquaculture nga species, mainly because yes, these are the prawns that are used in ebi tempura. So, it's quite a big enterprise, black tiger prawn. And what's concerning about GAV is that in the late 90s and the early 2000s, there have been outbreaks of GAV in Asia and also in Australia. So, there was a time nga wala nila na bantayan or like wala pa sila nakahibaw nga GAV dito, and exhibiting the symptoms as GAV, and then they called it mid-crop mortality syndrome. So actually they thought it was a complex of different viruses. It could be a complex of different viruses at that time, but they didn't quite get a grasp of it for katunga era. So, these are what they found out katong panahon na. So, they saw high mortality. Katong time it was 80%, and that is true of today. It is once affected, mortality rate is 80%. So, the prawns may congregate near the surface or the edges of water. As you can see sa picture here, murag naglubog na lang taon sila. They may start out as voracious, and then mukalit lang sila kawalagana. And then this is also typical of the disease and also YHV for that reason. Red ang body or appendages. You can see nga it's slightly red pod ang kaning nasa picture. Elahang feet and also parts of the tail. And lymphoid organ necrosis is also something to watch out for in GAV. The significance of this we will discuss later. They may become emaciated due to the anorexia, and their gills may be discolored. So, if you look at the picture on the left, you may see a sort of pale yellow na part sa black tiger prawn. That's the gills. Gill discoloration may also be a sign. They may have difficulty in taking oxygen due to the involvement of the virus. And you can also see a nice significant reddening jud sa legs. Black tiger prawns don't have red legs. And makita po diri sa right nga ang yang head nga part napoy reddening legs, and then also the tail. This is a significant reddening. So, naay mga microscopic na changes as well. On the left, you can see lymphoid, mo ang lymphoid organ. So, in the shrimp, the lymphoid organ may also be called the oca, which is why gitawag ang virus nga genus nga oca virus kay mo affect siya sa lymphoid organ. It's the etymology of the term is from Asia. So, you can see kaning mga vacuolation and also murag na hypertrophy ang iyang nucleus. And on the right, you can see pictures of the gills. There are some parts nga pycnotic or kanang mag-small na ang uh, iyang nucleus approaching cell death and karyopexis. Karyopexis, yeah, kani. Okay, next. So, in this, for the purposes of discussion, we shall explain the pathogenesis or pathophysiology in terms of horizontal transmission. But there are evidences nga ang GAV mo transmit siya vertically as well. So, in horizontal transmission, in the water, or for example, if maka-consume siya infected stock, ang shrimp, will replicate ang virus in the oca. So, mo ni ang predilection site niya, primary predilection site. And then, there will be inclusion bodies found in the cell cytoplasm. So, in the case of this virus, wala ko kakita og like specific nga type of inclusion body which is stated, pero it's basically mga basophilic, cytoplasmic, spherical nga inclusion body. So, with this, multiple organs may be affected. So, dysfunctional aside sa kanang lymphoid organ, ang gill, subcutis, nerve ganglia, mga gonads, and antennal glands, hemocytes, and hematopoietic tissues. So, after this, ang virus maabot na siya sa may appendages or like sa body surfaces nga makita ang reddening. So, reddening is caused by carotene nga pigment called astaxanthin in shrimp or prawn. So, it may be caused by multiple factors, pero sa akong nabasahan, it is triggered by cell metabolism or kanang stress response nga naa na ang virus sa iyahang cell mo nang mo express ang iyahang astaxanthin or ang reddening or marag orange. So, surfacing behavior happens when kana ma-affect na iyang gills. So, maglisod na siya og kanang breathe. So, murag dito na lang siya sa may kanang edges or sa surface sa pond. So, because they are so preoccupied with trying to breathe, they are not that interested in eating. So, magka-anorexia. And then, unfortunately, mamatay sila, 80% of them. So, it's quite a high mortality. So, in diagnosing GAV, you have to find the clinical signs. So, as I've mentioned, parehas-parehas sila og kanang signs sa YHV. So, you may need to differentiate those viruses, if you can, with RT-PCR. You may also do histology dito sa may predilection sites like sa lymphoid organ. You can do electron microscopy. You can do immunoblot and in situ hybridization tests. Wala lagi ni na italic. Doing in situ hybridization tests. So after that, kung discussing about the treatment, there are no treatment approaches available. So we can't vaccinate them as well. So vaccination isn't available, no treatments are available, mainly because it's a viral disease. So the best we can do is prevent it by doing disinfection. So we can disinfect the water by either heating it, usually at a temperature above 60 degrees Celsius, and then we can also use chlorine at 30 parts per million. We can also ensure nga wala jud ang GAV by buying specific pathogen-free or SPF nga mga stock. So this can also be verified if SPF ba jud siya by RT-PCR. And then, of course, we must ensure the water supplies are confirmed to be virus-free or kanang sourcing water not from kanang mga farms nga na-confirmed nga cases. So, prawns can be prawns may also be cannibals. So transmission can also occur by them by either sa water ra jud ang virus or kanang mangaon sila og other shrimp nga um, naa ana nga disease. So in stocks and sa water we must be careful nga gab free jud siya. So here are my references. Thank you so much.